Hello and welcome back to the NWR Virtual Healthcare Conference. My name's Matt Wright. Next up today, we have Vectus Biosystems, which is listed under the ASX ticker of VBS. And joining us to present on behalf of Vectus today is the CEO, Dr. Karen Duggan. Before we get started, just another reminder, if anyone in the audience has any questions, please feel free to type them in using the Q&A function within Zoom, and I will get to them at the end of the presentation. Karen, I'll hand it over to you. Please go ahead.
Thanks, Matt. Just to introduce Vectus, we're talk about ourselves as an anti-fibrotic drug discovery company. We have 3 first-in-class anti-fibrotics, which address major unmet medical needs. Our lead compound, which is in phase 1 trials, is VB004. We have a fairly exceptional patent portfolio encompassing more than 1,000 compounds, and this is about to be we're looking to extend that, and we have made 004 in significant amounts such that we can be discussing its clinical use. So to talk about fibrosis a bit, it is the replacement of functional tissue in organs such as the heart, liver, kidney, and lungs by scar tissue. If you get enough replacement, you end up with failure of those organs, and it is responsible for 40% of all deaths worldwide. So it's a major problem. And just to talk about our pharma, sort of what pharma would like us to achieve, a validated target. So what are we basing our therapy upon? Platform technology, by that they mean treats more than one organ system. Transformational agent, can you reverse existing disease? i.e., do you provide a hope of actual cure? Demonstrated efficacy in animal models, safety in IND tox studies. Can we make it reasonably cheaply? And what the cost of goods, can you make it, what the cost of goods is? Does our IP cover compositions of matter? Do we actually own the compounds as opposed to just the method for using them? How long are the patent lives? And then we are looking at phase 1 for human safety data, and then efficacy data. And this just shows you what we've already completed and what's currently in progress. So just to run through it, a validated target, we've shown that increasing VIP in the tissues, vasoactive intestinal peptide, in tissues such as the heart and the kidney, will reverse existing damage. So our target is replacement of VIP or with either by itself or a surrogate. And VIP itself is not amenable as a drug. It has to be injected, it's a peptide. So we went on to make actual mimics of it that you can take orally, that are small molecules. And 004, our first lead, looking at it in heart failure, we look at the damage in the heart when we start the treatment, damage in the heart when we finish the treatment. You can see it gets worse, and 004 in a dose-related fashion at the highest dose will reverse this existing damage. So in the histology, we see fibrosis as blue or cyano in color, starts off around blood vessels, extends between the muscle fibers. That starts to disrupt their capacity to work together and act as a pump. It gets worse over the 4 weeks of the study period, and you can see quite thick bands surrounding muscle fibers and starting to strangle them. With treatment, you have normal heart muscle. Similarly, in the kidney, we looked at the form of fibrosis, tubulointerstitial fibrosis, which is what puts you onto dialysis. Again, looking at start and finish, and you can see that 0004, again, the dose effect, it's better at all doses than in the heart. But again, we have this reversal of existing damage. What we're talking about is fibrosis that starts off partially surrounding some, but not all tubules, progressing to surround completely most of the tubules. And then with treatment, there is no fibrosis left around the tubules. So we've restored capacity for normal function. Similarly, we looked in the lung using a fairly nasty anti-cancer drug called bleomycin. It can, in fact, stop your cancer treatment because you wiped out your lungs. So again, 0004, looking at it start, finish, and we can see we've reversed the fibrotic damage that was present at the start. To look at it in the histology sections, you can see again cyano, the red dots are red blood cells in the very fine blood vessels called capillaries. And you can see you've got quite a lot of fibrosis and quite a lot of loss of capillaries, and that just gets worse over the 4-week period. So this is 2 weeks after we've given the bleomycin. This is 6 weeks after we've given the bleomycin. And then if we look at 0004, we've replaced all the capillaries, removed all that fibrous tissue. And the difference is that in appearance, a lot of it relates to the fact when you open the chest, the lungs collapse. But if it's got lots of fibrous tissue, it's going to sit there like it's made of plaster of Paris or cardboard. And so it appears to have some more air spaces than your normal tissue. Our synthesis program, we made first in New Zealand, a company called Glycosin. We made a 5 kilogram scale. Our second synthesis, we made 3 validation batches, which complies with FDA requirements. At the 5 kilogram scale, it costs 5 cents a milligram. So cost in synthesizing this drug is not an impediment to it going forward. Our phase 1 study, we just looked at a very conventional single ascending dose SAD, multiple ascending dose MAD. We included pharmacokinetic studies. We expected to find a maximum tolerated dose, maybe dose limiting toxicity if it's present, and get some pharmacokinetic data. We had Syneos Health monitoring the trial, and it was conducted at Nucleus Network in Melbourne. We completed the SAD with no significant adverse events. Our planned dose, top dose was 300 mg, and we achieved that. The repeated dose study of 14 days, we had a planned dose of 100 milligrams each day, and we completed to that dose with no significant adverse events. Our PK shows we reach maximum concentration at 6 to 8 hours with a half-life following that of 10 to 15 hours. Importantly, when we repeat it, we do it at the start, we repeat it at the finish of the 14 days, we have no accumulation. That is one of the problems. If you get accumulation, you may get off-target effects and side effects. So absolutely clean pharmacokinetics. This just shows you what it looks like. 6 to 8 hours for the maximum concentration in blood. And then at 10 to 15 hours later, you can see you've reduced that concentration by 50%. That's your half-life. So 0004 is very safe. We achieved the maximum planned doses in both the SAD and the MAD, and our PK is consistent with once daily dosing. So in summary, it's a VIP agonist, and it is first in class. It's transformational. We can make it easily, and it's cost effective. Our stability study showed that it's stable in excess of 2 years. And in terms of animal studies, the efficacy is much greater. in the diseases we've studied than currently available agents. So we think it may, in fact, replace a lot of those as first-line therapy. Sorry, I need to go back one. Sorry, I've gone too far and I can't get back. That was the previous slide, was just to show you that we're at the point where most trade deals are done with pharma, and gives you an idea of valuation at that point in time, at that inflection point, and we are roughly there. So continuing on, we have 0004 and what we think we'll be seeing this year. 0004, hopefully completing the phase 1B human trial, which is currently in progress. Talking with that data to pharmaceutical companies. A32, VB4A32 will start the GMP synthesis, and once that's completed, look at IND tox studies. We are accelerating work on other compounds. We are moving to new premises where we've outgrown our current ones, and we're working through our library in terms of Alzheimer's, and we have some commercialization work happening with Accugen. No, it's not letting me. So this is just showing you the VB4A32 data, where again we see a dose response curve and reductions in fibrosis that were present in the liver compared with the starting point control. This just shows you what the fibrotic damage would look like around blood vessels and extending out between the liver sinusoids, and we restore normal sinusoidal architecture with 6 weeks treatment. Some new stuff we've been doing has been looking at Alzheimer's, and we believe that it's a protein deposition disease, in this case amyloid plaques. These disrupt communication between neurons, and you develop what are thought to be neurofibrillary tangles within the cells, the brain cells, with a protein called tau. The combined effect is that the neurons die and you get the shrunken brain. And so we've a new group of compounds, the Gen Zs, which we've been testing in an in vitro system which models Alzheimer's, looking at fibril digestion and removal of intracellular tau. So this, we use a protein fibril in this, which is much more resistant in this system to digestion than amyloid beta would be. And it's a recognized model because of that. And what we can see is that at the red bars, we actually speed up the enzyme digestion of this protein. So it's suggesting our drugs would have a role in helping digestion of the amyloid protein, and then looking at the intracellular tau protein. This is what destroys the neurons, or thought to destroy the neurons. And you can see with treatment, we remove tau protein from the cells and move it to the extracellular space, which would be the CSF in vivo, and that would allow the tau to then migrate out and be excreted like any other protein. So we believe that what we have is something that not only does something about amyloid accumulation, but has the capacity to do something about the tau protein problems, which the current therapies only treat the amyloid problem and have no effect on the tau pathology. So we believe this is new and will be quite exciting. Okay, I guess that means we've moved to protein deposition diseases from being just a fibrosis anti-fibrotic drug discovery group. So we've got two groups of protein deposition diseases, the fibrosis related stuff, which is collagen and fibronectin, and we see that underlying heart failure, kidney failure, liver, cirrhosis and pulmonary fibrosis. And as I said, accounts for 40% of all deaths. And then we have the amyloidoses. Accumulated proteins do vary. In the brain, it's amyloid beta, and you get Alzheimer's in the heart, you get transthyretin as the protein, and you get cardiac amyloidosis, which leads on to a cardiomyopathy. TTR depositing in the kidney gives you renal amyloidosis and ultimately renal failure. You can get problems in the intestine and malabsorption with amyloid A, beta-2 microglobulin or prealbumin deposition, and in the pancreas, islet cell amyloid protein, polypeptide, can deposit, destroying the islet cells. And this is the transition in type 2 diabetes from being able to take drugs or needing insulin. These are mostly rare diseases, while the fibrotic diseases are quite common. So removing protein deposits that cause disease is really a major unmet medical need, and that really is Vectus's territory. This just reminds you of our very large patent portfolio with good patent life on all of them. And that's it. Thank you. Matt.
Thanks very much for that presentation, Karen. We'll move on to some questions. Just as a reminder, if anyone in the audience has a question, please feel free to type it in using the Q&A panel within Zoom. Karen, I'll start off with, I know you probably can't say too much, I need to be a bit careful about how much you say, but can you just provide some colour around discussion with potential pharma partners that you've had today?
I can't really say who they are. That probably would be a deal breaker. So we have had engagement with both we are engaged with both major pharma and mid-sized, more specialist pharmas that, you know, might be specializing in lung disease or liver disease, those sorts of things, as well as the larger pharma that engage across all platforms.
Excellent. And the Alzheimer's program that you talked to towards the end, what's next? What are the next steps there, and what needs to happen to get that in?
Okay. That's our first pass data. As I alluded to, we have a new group of compounds. Our first bit will be to look at those and see which of them is best. Whilst we patent a lot, obviously. So accumulation of data and then moving to animal models when we have a lead compound, and then taking it through the rest of the trajectory of, ion synthesis, ion detox, etc. But the first step is just working out which of these new compounds will be the lead compound.
Excellent. Karen, if you just try on your keyboard to press left or up just to go, can you go back back on slides or not?
Should be able to.
Yeah, someone's just asked if you can go back to 14. I think that was the slide that you might have gone over, skipped past.
Right, there we go. Is that it?
Yeah, did you just want to give a bit of
right,
color
okay.
on that one, please?
Yeah, I wasn't being able to go back while I was actually in the talk. It seemed to jam. Okay, so this just sort of shows where most of the deals have been done, and you can see that it's around this sort of phase 1 to completed phase 1 stage, and we'd be here at the moment. We've sort of completed a 1A. We're working on the 1B, And so what you see is a lot of pharma deals, very little preclinically, pharma deals coming in around this space, less so around the phase 2s, and then again a bit more around the registration licensing stage. But what you can see is significant inflection in market value or value for the drugs and the companies at that point in time. And that's what we're hoping to achieve with our discussions with pharma. So you'd be looking at these reflect, sometimes it is the sale of the compound and outright, and that's it. In others, it is a upfront payment and then a series of milestone payments leading up to final price on licensing, and the milestones adding up to that valuation at licensing.
Yeah, no, that makes sense. Thank you for covering that one. And just another favourite question, which is just in terms of news flow through the rest of the year, just at a high level, what should investors be looking for in terms of news flow from the company?
Okay, news flow will be stages through the 1B, you know, patient enrollment, patient completion, data lock, and then reports. And contracts for A32 synthesis once we've finished negotiating those, and stages along the way of you know, batches made, those sorts of things. All sounds a bit tedious, but you know, it's all part of the grist that you can actually make the drug. And then obviously in terms of the new compounds, we'll be announcing when the patents are lodged. We can't do much more than just communicate that we have some data and are moving there with new compounds.
Well, that sounds good. And a question that relates to this slide when you spoke before, Karen, that's come through is what type of deal would you say is likely with pharma companies, as in would it be Vectus licensing the use of VB004 or selling it outright?
Our preference, and I'll state it that way, because, you know, there's always a deal that's too good to refuse. But our preference would be a licensing deal, upfront payment, milestone payment, and then royalty on licensing, on actual marketing.
That sounds good. Thank you very much for the presentation, Karen. That's all the questions that have come through. And thanks to everyone for joining. We've got a few presentations left today. Next up is Paradigm Biopharmaceuticals in about 15 minutes. But for now, thanks, Karen, again for the presentation.
Okay, thank you. Bye.